Kan je me nou horen?
Ja, nu hoor ik je wel.
Ik begrijp er helemaal niks van. Ik moet me dus elke keer weer opnieuw inloggen.
Ja, dat weet ik ook niet, die magie van die dingen.
Nou ja, ik probeer het ondertussen wel. Dus ik hoop dat we straks kunnen beginnen.
Ik heb net beseft, inderdaad, ik ga die vragen in de chat niet zien verschijnen, denk ik, want het scherm gaat geblokkeerd zijn.
Ja.
Dus ja, jij moet maar zeggen. Je mag mij tijdens de presentatie ook even onderbreken, hoor. Misschien is dat nog handiger. Maar je ziet maar hoe je het aanpakt.
Ja, nou ja, geef maar een break aan. En ik denk dat we dat gezamenlijk wel heen krijgen.
Mocht je nog een keer wegvallen, Gertrude, probeer anders je camera uit te zetten en alleen je microfoon aan.
Ja, dat doe ik ook zo. Ja, dank je voor de tip, want ik denk dat het dus een internetprobleem is, vermoed ik.
Ja.
Connectie. Even kijken, ik zie geen enkele attendees op dit moment. Ik moet heel even kijken of ik die kan zien.
Ja, de room is nu open. Ja. En ik heb de opname al vast gestart.
Die heb ik gezien, dank je. Ik ga straks inderdaad mijn sowieso muten en mijn microfoon uitzetten. En dan hoop ik ook ik ga ervan uit dat het netwerkproblemen zijn die ik daar heb. En als ik wegval, dan log ik me gewoon weer opnieuw in.
Ja, prima.
Okay, so everybody, welcome to our very first session from 2022. I can say good day because for some people it will be in the morning and for other people it will be afternoon. And I'm very happy to welcome you. And the first talk that we are joining at this moment is from Peter Dedecker, coming from Leuven, Belgium, talking about his research and the research he's doing with his group on chemistry of fluorophores. And as all of you know, is that fluorophores are one of the main ways of interrogating all our experimental subjects, including cells and tissues. So this is a very important part. And Peter, please continue. And also, for you as attendees, when you have questions, please put them in the chat. And we do it like this, that when there is enough questions and when there is a certain subject that Peter kind of finished, we will address your questions that you posted in the chat so that we can deal with them. So this is how we kind of structure this very, let's say these next oncoming 2 hours. We will put them in parts. Please place your questions in the chat, and we will address them according when there is a kind of chapter closed within the talk. So Peter, the stage is yours.
All right. Good morning, everyone. Thank you, Gertrude, for the introduction. So welcome indeed to this tutorial session that's going to be mainly about labeling and labeling techniques. Now, I should say that I'm very much flying blind in the sense that all that I can see are my slides. I can't really see the chat window or any of the participants. So I'm really talking to my laptop. But indeed, the plan for this tutorial is to also invite discussion. So please, if you have questions at any time, ask them via the chat. That would be nice. And then Gertrude will pick them up. Unfortunately, I won't be responding directly because I can't see any of those at all. Good. So in any case, welcome to this tutorial. So we will be talking about fluorescence microscopy, which I assume many of you want to use because of the many nice advantages that it has. And the goal really is to talk about labeling and fluorophores, which are, of course, essential parts of that. So this is a tutorial. So my goal is to really start a bit from the basics and give you an overview of what is really a very fundamental and a very broad field. So unfortunately, I won't be able to pick up on all of the richness that this field has. I'll be trying to give you a bit of the highlights, starting from a very basic level. And again, any questions are absolutely welcome. Just ask them in the chat and keep in mind that I won't be seeing them directly. All right, so what am I going to, or what am I hoping to cover as part of this tutorial? I'll start by talking a bit about the chemist's view of fluorescence, just to get us all started from the same page. Then I'll talk a bit about common labels that we are using, common fluorophores that we are using. I'm going to focus on labels that see a lot of use. And that also means, unfortunately, that I'll have to exclude a few less common types of labels. I'll talk then about labeling methodologies, the question of how do you get the label to where it has to go, the target that you want to visualize. So I'll also try to give an overview of those. Then I'll talk a bit about the smart labels, or quite a bit about the smart labels that I've promised in the title. I'll tell you a bit about what those are and how they can be useful. And then finally, at the end of the tutorial, I'll highlight some notable trends and developments that are ongoing. Some that selected that I think are really driving the field forward. Of course, there are other developments as well, but I'll limit myself to a couple. Overall, I only have materials for about, say, an hour and 15 or so. So really, it would be nice to have a discussion as well about anything you want. I deliberately left space within this slot to do that. All right, so let's start with the basics, the chemist view of fluorescence. We all have a basic notion, I think, of what fluorescence is, but from a chemical perspective, it's really an interaction of light with molecules such as quinine, which is the first fluorophore, actually the fluorophore that was first used to observe fluorescence on back in the 19th century. And from a molecular structure perspective, one feature that you will always have with fluorophores is that you will have a presence of so-called conjugated systems. These are delocalized electrons. Typically, what it means is that in a fluorophore, you will see aromatic rings and you will see double bonds all forming an extended system. And this is what you really need to absorb light within the visible wavelength. Now, moving forward, we know that the energy levels of such molecules, or all molecules, are quantized. We know this based, for instance, on experiments on the emission of hydrogen gas, where people discovered that you only get discrete lines in the emission spectrum. And what this really showed is that the energy levels in all molecules are quantized. And this is a very typical way of representing those. Here we have different levels in which the molecule can exist. And we have increasing energies available, or increasingly higher energy levels available. The lowest energy state we typically call the ground state. The higher energy states we will call the excited states. And on top of all of the different electronic levels, we also have sub-levels that essentially consist of states in which the molecule has different degrees of vibrational energy. So what happens when light is absorbed, when light hits a molecule, and the wavelength of the light is such that it corresponds to an energy difference between two different levels, then the photon can be absorbed, which means that its energy is transferred to the molecule and the molecule enters a higher excited state. Typically, what happens then is that some amount, the vibrational component of this energy is lost as heat, so is given away as heat. Which leaves us with a molecule in the excited state. Typically, the energy that it acquires in this way is quite high. So the molecule will tend to get rid of it quickly. And one way in which it can do this is to emit a photon, and then we end up with fluorescence emission. The timescales of these phenomena can be very different. Absorption is a very fast process occurring within femtoseconds. Fluorescence emission is much, much slower, typically occurring within the nanosecond regime. So one molecular insight that we learn from this is that the absorption and the emission spectra are really determined by the energy levels that are available within the particular molecule as such. The fluorescence process has to compete with other molecular events. One of these is internal conversion, meaning that the molecule loses energy as heat. And of course, if the molecule loses energy as heat, then it can't very well emit that photon anymore. So there is a competition for the processes that happen in the excited state, and there is a certain probability that fluorescence will happen. So this scheme gives us a number of key insights. This scheme is known as a Jablonski scheme. It gives us a number of key insights. namely that absorption and emission spectra are determined by the molecular energy levels. So the distance between the energy levels determines the absorption and emission spectra. And this is, of course, determined by the molecular structure. And as I mentioned before, in a fluorophore, you will typically have an aromatic or a conjugated system consisting of delocalized electrons. And the rule of thumb is that as your system of delocalized electrons gets bigger, you actually decrease the spacing between these levels. And so you end up with more redshifted emitters. But this will become more apparent later on in the talk. A second insight is that the ease by which light is absorbed is described by the extinction coefficient. So the higher the extinction coefficient, the more light or the more easily light will be absorbed. And the extinction coefficient is really determined by the quantum chemical nature of the molecule, including things such as overlap of the wave functions, symmetry, and also spin considerations. But as a rule of thumb, we simply remember that a higher extinction coefficient means that a transition is more allowed and thereby is more easily excited. And fluorescence is just one of the many processes that can happen from the excited state. And so it always competes with other processes. The chance that a molecule will emit a photon is known as the fluorescence quantum yield. So as the quantum yield gets higher, there's a higher probability to get a photon. So bright dyes have high extinction coefficients, meaning that they're easily excited, but they also have high fluorescence quantum yields, meaning that after excitation, they are likely to emit a photon to lose the energy that has been acquired. Now, if you look at such Jablonski schemes in books or publications, you will typically find more complex schemes involving multiple transitions. And what this really reflects is the richness of processes and states that can occur. One that you often see is singlet states versus triplet states. That has to do with the spins of the electrons. In the wave function, typical dyes, I should say, that we use, organic dyes, are typically in the singlet states. Occasionally, they can cross to triplet states, which have a number of different processes. But what this really highlights is, that the excited state can use its energy in a number of different ways, such as losing it as heat, emitting a fluorescence photon, going to states with different spin multiplicities, cis-trans isomerization or rotation around bonds within the molecule, photochemistry, and these give rise to other derived processes. For instance, intersystem crossing, formation of the triplet state, causes your molecule to become non-fluorescent for a period of microseconds to milliseconds. But it can also lead to events such as formation of reactive oxygen species, meaning that you create molecules such as singlet oxygen that can cause damage to your system by being highly reactive. Also, we know that all dyes undergo photochemistry as well, because we know that after a dye has performed a number of excitation cycles, it will photodestruct. So it will become destroyed and not be fluorescent anymore. And what this really reflects is the fact that upon absorbing a photon, you acquire a lot of energy, and you can use this energy to do various things that otherwise would not happen. One nice thing about these considerations is that, first of all, most molecules do not absorb visible light because they lack a sufficiently large conjugated system. And so most molecules lack conjugated systems that are big enough to give them an absorption within visible light. And second of all, the vast majority of molecules relaxes too quickly for fluorescence to occur. So it's very unusual to have molecules that both have good visible light absorption and molecules that then also can emit fluorescence photons, which is why fluorescence is so selective. So one of the key features of fluorescence microscopy that we all know and love really arise because of the chemistry involved in the process, which says that not many molecules are capable of bright fluorescence emission. Okay, Gertrude, any questions about this part?
No, I haven't seen some. Can you hear me?
Yes, I can hear you. Okay.
So I haven't seen any questions. So I think I would continue because I think we might need a little bit deeper information. But yeah, I think it's a very good introduction. But
yeah. Okay, moving on. Obviously, a key consideration in fluorescence microscopy is to have proper labels that can stain the features that we would like. So the tools of the trade, or the labels that we have, really consist of a number of different families. On the one hand, we have synthetic dyes that historically were the first labels to be used, and that really consist typically of molecules that are synthesized by teams of chemists. The second class of significant labels are fluorescent proteins, which obviously we all know and love as well. Then we have more exotic systems, between quotes, such as quantum dots, nanodiamonds, and the like, systems consisting of metals and the like as well. And finally, we have a fourth class of labels, which is now really emerging over the last couple of years, which are hybrid systems between these synthetic dyes and the fluorescent proteins. So in this talk, I'll be focusing a bit on the dyes, the fluorescent proteins, and the chemogenetic systems. I'm not really going to talk about the more exotic systems, as I call them here, not because they are not interesting or not because they are not useful, but simply because I think the vast majority of experiments really works based on the classes 1, 2, and 4 that I have here on this slide. So let's talk a bit about synthetic dyes. In this perspective, you have a very nice overview of some of the common dyes that are used. Here we have fluorescent proteins. I'll talk more about these later. What I'd like to talk about now are really these particular molecules here. And they really span the visible spectrum, going from UV and violet all the way to the deep red or even the close infrared. And one of the things that you may notice, first of all, is that a lot of the molecules have very similar basic structures. And indeed, the basic layouts of many of these molecules have been known for a long, long time, in some cases already in the 19th century. And so you have frameworks such as coumarines, fluoresceins, rhodamines, carbocyanines, body-P dyes as well. These are molecules that have chromophores similar to the fluorescent proteins, pyrene dyes, etc. Overall, we have a limited number of families of dyes, and most or nearly all of the dyes that we use actually originate from that limited number of frameworks in use. You can also see here what I was talking about before, that in general, more blue shifted dyes have smaller delocalized electron system conjugated systems, but red dyes typically have very large or comparatively much larger delocalized systems in use. So all of the dyes that we use more or less come from a number of these typical families. And to a large extent, a lot of the dye engineering that has been done has come down to making specific modifications from these basic molecular scaffolds. And that includes things such as making them water soluble by adding charged groups. For instance, here you see a sulfone group. That is really the defining feature of the Alexa family of dyes. Other attempts have been to include more rigid elements to shift the spectra, but also try to increase the quantum yield. Other engineering attempts that have been done are introducing heteroatoms to change the molecular properties. But by and large, really the dyes that we use today are modified versions or evolved versions of frameworks that have been known for quite a while already. So just to highlight this a bit, this is an example where we have the basic carbocyanine dye scaffold. And what you see is differences arising in the addition of charge groups. So these charge groups are typically used to make the dye water soluble, because you have, in this case, fairly large, often hydrophobic systems. So the addition of charges really leads to having a water soluble dye, and can also help with the conjugation. Other changes are rigidifying the dye. Here you see that the groups have been added to make the dye more rigid, to increase its brightness. Here we have a number of different modifications. You see alkylation happening, heteroatoms, etc., etc. One thing that I can maybe point out is this is the extinction coefficient. This last number is the quantum yield. Typically, dyes more in the blue have lower extinction coefficients, but high fluorescence quantum yields. Dyes in the red typically have higher extinction coefficients, but lower fluorescence quantum yield, which arises because they have because of basically the intrinsic nature of emission in quantum mechanics. Other modifications, again, focus on solubility. I've told you about addition of charge groups. One of the downsides of this is that charge groups make it hard for a dye to cross into over lipid membranes. So other modifications have been followed as well to try to get dyes with better properties. So key challenges typically are, okay, you have a dye. It is fluorescent. How do I get it inside cells? How can I get it to pass the membrane? Or do I need to permeabilize the cell? And once it is inside the cell, how do I obtain targeted labeling? And often this requires clever chemistry. Here's a paper where we have a dye, silicon rhodamine, which actually can exist in an equilibrium between a charged and an uncharged species. And so what happens is that the uncharged species can make it across the membrane barrier. But then the charged species actually then becomes fluorescent. So a lot of the engineering of dyes has been driven by the need to get the dye into cells and also the need to try to avoid background staining. So these are typically prime concerns. Here's an example of a range of dyes where you find that for some dyes you see significant background staining. So these are all considerations that drive dye development. The second big class of dyes is fluorescent proteins. Now we all know and love, or at least I know and love fluorescent proteins. I know and love organic dyes as well. But just as a very brief introduction, so these are proteins of around 25 to 30 kilodaltons, and they are really genetically encoded fluorescents. So when the cell makes these proteins, they will fold into this very characteristic structure, and you will obtain a chromophore that will be fluorescent within the center of this structure. And the chromophore is really formed fully automatically from the amino acids present at that location. So no intervention is needed, apart from a need to consume molecular oxygen. Now we know by now that there's a very broad range of fluorescent proteins available. Typically, and this is a bit older data, but typically purely optically, the green and the yellow fluorescent proteins tend to have the highest performance. So some of the key goals in protein engineering has been to try to expand this palette as much as possible. And what is intriguing also with these systems is that all of this diversity really arises from just a handful of chromophores. So there's only a small handful of chromophores that give us all of this diversity. And as you can see, they are all formed through chemical processes occurring within the intact protein itself without further intervention needed. Another element that is intriguing is that at a macro level, or sort of at a high level perspective, they are all very, very similar. So this is an alignment of over 100 fluorescent protein crystal structure. By and large, they are very, very conserved in terms of structuring. Now, as we know, these allow us to do very nice things, such as the genetically encoded labeling of large organisms, or neural development and the like, or even some kind of fluorescent PETs that people have been trying to make. This is fantastic. There's also a few things about fluorescent proteins that are less well known. And part of that is that they actually have a very rich photochemistry. So if you look, for instance, at a single molecule trace of a fluorescent protein, then you see that even within emissive periods, and this is a special protein called a photochromic label, but what you see here, for instance, within that single molecule, is that within the emission, you have a lot of diversity going on. You have a lot of dynamics going on. So they really have a very, very rich photochemistry that can be an advantage, but can also be a disadvantage in the experiment. A second element of fluorescent proteins is that they may not all mature successfully. So the chromophore is in principle formed automatically, but this has a number of restrictions associated with it. And actually, you can readily see that, for instance, here we have E. coli bacteria expressing different kinds of fluorescent proteins. You can see that depending on temperatures, different variants do not become fluorescent to the same degree. And what this really tells us is that fluorescent proteins occasionally can have trouble forming a chromophore, which depends on how they are used and also which label is being used. Other issues that can arise with fluorescent proteins are issues where they interfere with the proper cellular functioning of the cell. For instance, this is a paper where they took proteins and just fused them with different fluorescent proteins. And they ended up having completely different cellular distributions, reflecting that the label can indeed interfere with the chemistry. This is another example where there is a protein involved in the E. coli cytoskeleton that was long assumed to have sort of a helicoidal shape within the cell, a bit of a helix. Based on what people had seen with fluorescence microscopy. But actually, it turned out that this was purely a result of the tagging that was being used, and then that if you change that tagging, that people saw a completely different distribution that was consistent with electron microscopy data. So overall, fluorophores have a number of good, bad, and ugly elements to them. Starting with organic dyes, really, the very nice elements of organic dyes are they're small, and they have exceptional optical properties. And I will come back to that later as well, but they have an extremely large spectral tunability. They can have very high brightness, high photostability. From an optical sense, they are really very high performing. They also have a bit of less positive elements, which is usually questions around how do I get them into cells? How do I get them into big systems? What if I have many cells? Specificity of the labeling, labeling completeness. What happens when I have excess dye? What do I see? Will that perturb my measurement? And also issues surrounding long-term observation. If I have a system that is alive and that I want to monitor over hours or longer, how do I do that given that really the dye only is there because I have to manually add it? It also has an ugly element, which is that, of course, you have to buy these dyes, and that can have, let's say, that can produce a bit of a barrier to entry. Fluorescent proteins, on the other hand, really shine because of their genetically encoded nature. They have a reasonable spectral tunability. They give you essentially a perfect labeling because you know that every protein that is expressed that is fused with an FP will have a copy of the FP. And it will be in perfect stoichiometry. And this also means that long-term observation is easy because the label is being produced continuously. The cost of such a label is also approximately zero, which is in fact probably a key reason for why fluorescent proteins have, or one of the key reasons for why fluorescent proteins have made such an impact on the life sciences and the use of fluorescence microscopy. Less positive is that they are large molecules, which also means that they have a large interaction surface. So they can interact with many things in the cells in theory. And in general, their optical properties are much less favorable than those of organic dyes in terms of brightness, stability, spectral tunability. Those are all considerably worse. They have complex photophysics and photochemistry, which can be an advantage or a disadvantage. And they also have various environmental sensitivities. I will come back to these topics later on. And then ugly is always the question of, okay, how many of the proteins that are really expressed really do become fluorescent? Now, just to give you an idea of the differences in optical performance between organic dyes and fluorescent proteins. So this is an assay where we tried to measure photodestruction. So we had cells that expressed fluorescent proteins and other cells that expressed a range of organic dyes. And we simply irradiated these cells at about the same excitation rate. So say X number of excitation cycles per second. And as you can see, fluorescent proteins tend to bleach very quickly, tend to photodestruct very quickly. And with organic dyes, you can actually push this over a much longer duration. Though again, it very much depends on the nature of the organic dye. So some dyes have a much higher resistance than others. And this is something that often is not very well understood. All right, so that's it for the introduction of the different types of labels that we will be considering. We will be considering a few more, but at least the two basic families. So I'll just have a little pause here if there are any questions about any of this part.
Okay, hi, thank you, Peter. At the moment, I don't see any questions, and I would like the audience to paste any questions in the chat box so that we can address them. In the meantime, Peter, thank you so much for this overview already, and also for showing, let's say, the advantages, disadvantages of TFPs and soluble molecules, because this is something that we really often have to address and also to take in consideration. What I've been wondering about, I used a lot of resin proteins in the past, and I now kind of switched to using organic dyes. And sometimes I'm really troubling and solving solubility of the dyes. And also, so could you maybe, the first question I'm having is, could you maybe tell a bit more about solubility and how we could solve that in the lab when we have we bought such a fluorophore, so for such a large amount of money, and then it starts to precipitate. And the second question I'm having is, I also started to notice that most of the time I have this more with infrared dyes in comparison to visible dyes. Maybe they are already better developed and designed in the meantime, but is there anything I can do there in that respect?
Yes. Well, it's intrinsically a tricky problem, I would say. And maybe it helps for a moment to come back to the overview of the different dyes. Maybe first of all, the question relating the infrared dyes, solubility often has to do there with the fact that you end up with large conjugated systems that have a tendency or a propensity even to potentially aggregate or have a significant hydrophobic character. So the fact that you end up seeing aggregation more with with infrared dyes is not entirely unexpected, let's say. Driving the solubility in an aqueous solvent in water is really mainly determined by the polarity of the dye. So what this means is that this is a key feature that people try to optimize when they do organic dye synthesis, for instance, by adding charged groups to prevent the dye from or to give it a good water solubility. There's not really all that much that you can do to affect this. To a large degree, it's encoded in the structure. You can consider exploring different dyes. Concentrations, of course, help. So you need to carefully tweak concentrations, try to have a fairly low concentrations if possible, experiment with this, titrate it a bit. Occasionally, the delivery vehicle may help as well. So some tricks are to, say, apply dyes in the MSO, but then you can have also issues with cell viability being affected. By and large, it's a tricky consideration. Part of the trickiness is also that if you want to get the dye inside cells, or at least across plasma membranes, then actually you don't want it to have all that money, or you don't want it to be net charged, because then it won't cross the membrane anymore. So I think what you're really seeing are the different trade-off spaces that people have when they design organic dyes, and the trade-off between cell permeability, water solubility, and this is something that really depends on every dye structure, and it's sort of tricky to actively prevent this yourself. I think the easiest way is to be very careful about the concentrations that you apply and maybe consider a delivery vehicle. Infrared dyes, yes, can be more challenging because of their molecular structure, essentially.
Okay, thank you. In the meantime, there are questions on the chat that I would like to read aloud for you. And the first question comes from Rafael Picard, and he has a question about one of the last lifetime measurement slides. So lifetime measurement on last slide, so maybe you can go there.
So probably the photostability, I take it.
Yes.
Yeah, so I will go there.
Yes.
Voila.
Yeah, exactly. Because his question is, for the organic dyes, it looked like the signal to noise ratio was higher. And he asked himself, is there any reason for that?
Yeah, the signal to noise ratio here being lower, I take it, right? Because if I look at the organic dyes, I see a bit more noise there. I would have to go back to these experiments to see. I think it has to do with a number of elements. First of all, these are experiments where we have cells in dishes. So we had here HeLa cells or the like. I'm sorry, I can't remember which cell line we used. But we have cells expressing either fluorescent proteins directly or SNAP and halo motifs, which I will come back to later. And then these are being stained with organic dyes or with FPs. And to a reasonable approximation, the amount of noise tells us something about the brightness that we saw for those cells. Now, first of all, as a disclaimer, you should take this with a bit of a grain of salt. Because of course this also depends on the light sources and the filter sets that we have, first of all, how much light we can collect. But we also see fairly significant variability in staining, and we find that some dyes are harder to stain than others, and this is something that also we try to optimize a bit. We're now actually better at it, I do have to remark, but yes, to be fully honest, the higher noise here means lower brightness for the cells, which can be due to different expression, but also to a significant degree reflects different degrees of labeling efficiency.
Thank you. Next question comes from Vanessa Carlos. Her question is, what criteria would you suggest to consider for choosing FPs or organic dyes?
Basically, I'll come back to that later. Let me get back to it later. Okay, so I'll postpone, if I may, Vanessa. I'll postpone your question. There will be a slide about labeling advice later on, and then if that is not clear yet, then please raise your question again. Okay.
We do that. In the meantime, Raphael also answered, yes, he meant lower, so it makes sense. Thanks. Then I have a question from Laura Vitadello. What is the maximum wavelength which of that can be reached in infrared?
Yes, so actually I will have a slide about that more at the end of the talk. People have now been pushing dyes into beyond the 1000 nanometer window, even for direct excitation. So then you get into molecules that essentially have even longer conjugated systems, right? So you have here an even longer conjugated system. So those molecules exist. The downside is, or the challenge is in a number of different things. First of all, you may need different hardware to start seeing such long wavelength emission. So you're talking often different types of detectors that can be tricky to acquire, or at least specialized to acquire. And second of all, those dyes typically tend to have poor brightness. Because of the fact that as your energy levels gets closer, your rate constant for radiative relaxation gets lower and lower. So the answer to your question is that this is increasingly being pushed, and I'll have a slide about that later in the talk. But very often these result in images that are not like what you would expect, cells showing very bright features. Or subcellular structures, but really start to be more about measuring large masses within, say, lab animals or whatever. So there is no real upper bound beyond, or let me rephrase, the range here suggests that you can easily go into the near infrared, but what happens is that you start losing a lot of brightness, and so you end up with different types of measurements that often need different equipment. I think as a rule, people now are doing imaging with dyes up to excitation, say around 700 or so. This can be done, but it gets more and more challenging simply because it gets harder and harder to have a bright dye emitting at those wavelengths. Also with fluorescent proteins, not fully related, of course, but with fluorescent proteins, we typically have the brightest molecules around the green or yellow, and then tailing off towards the blue or red. But I'll have a separate slide about that later on.
Okay, then another question from Sabine Knip. Just wonder, is bleaching associated with an increase in heat emission?
Bleaching as in photo destruction. I don't, so it's a question that I've never talked about. I think the bleaching itself is a way, is a process. It may be intramolecular, it may be intermolecular, is a process where you end up doing some chemistry that ends up destroying your molecule. As to whether this chemistry would be exothermic or endothermic and whether that would be significant, I don't really have any specific numbers on that. I doubt that it would be really a strong heat-inducing effect because it's a one-time process, right? The molecule bleaches once within a simplified framework. But it is true that a lot of the acquired energy for any fluorophore, many excitation cycles will actually not deliver a photon at all. And there you will have heat being produced by the molecule. So yes, in principle, there is a heating going on. Whether there would be more by bleaching, I don't have the numbers, but I would be a bit surprised, to be honest.
Okay, and next question comes from Nicole. She says, individual fluorophore, Nicole Slesiona. So individual fluorophores with the same structure seem to have quite some variability in their brightness. If you look at bleaching traces over time, for example, Alexa 647, yeah, one fluorophore might show 3 times the brightness compared to the other. Do you know why that is? Is this just an effect of orientation in the light path?
So I'm not sure that I fully grasp the question, to be honest. Gertrude, feel free to correct me or the person asking the question. So first of all, changes in brightness. Yes, so fluorescence brightness can be difficult to predict. Small substituent changes, small molecular modifications can have a strong impact on molecular brightness. And really, as far as I know, there are no simple rules describing this. So people try to make some rationales for why certain substituents make changes. What happens when you have electron donating groups and the like present. But by and large, there are no really easy ways to predict how fluorescent something will be unless you have years of experience working in dye chemistry. Perhaps for me, I also find it tricky to predict which one of these will be brightly fluorescent. But then I didn't really catch the link with the orientation, to be honest.
Okay, so
sorry, please ask again later on,
yeah,
and then
Nicole,
we will rediscuss.
If your question is not, if you have, let's say, I hope Peter was possible to address your question as the question was stated. If not, please readdress. I have the question of Vanessa was already bookmarked for when, for the next session.
The advice on labeling, yes.
So my suggestion is we continue with your talk and we keep, I keep this question already.
Please. All right, and thanks for the questions, indeed.
Yep.
Okay, we have the tools of the trade. We have dyes, we have fluorescent proteins. How do we now label them? What are the ways to get them into cells? What do people tend to use nowadays? And I'll start with fluorescent proteins because fluorescent proteins to a large degree make life easy. Because typically we are interested, or very often we are interested in labeling proteins or protein-based components, or we have knowledge of protein machinery that will direct itself to the structure that we want. And so with fluorescent proteins, nearly always the way in which we label them is by making genetic fusions to the gene that we want to visualize, such that when the cell makes this, gene, when it expresses this gene, it will actually intrinsically become fluorescently labeled by having the protein attached. It doesn't have to be a full gene. It can, of course, also be a targeting motif that will direct the label somewhere to the plasma membrane, to the mitochondria, to the ER, to peroxisomes, etc., etc. But by and large, simple genetic fusions do the job very well. And they do it so well that with very little effort, you get live cell compatibility, you get perfect specificity, you get perfect labeling stoichiometry with the caveat of imperfect expression of the label. You get compatibility with all genomic tools. And that is a very strong element, of course, and with all future genomic tools that may be developed as well. And your label is made for free. You don't have to spend any effort, any optimization to some degree. The label, the hard work is done by the cell itself. And this is also key, this convenience in labeling, for instance, multicellular systems. Imagine having to put an organic dye inside such a system. That would actually not be all that much fun, in my opinion. But with a fluorescent protein, you sort of get all of that for free. Actually, you might be curious about how easy is it to attach labels. Do you perturb something? Actually, most or many proteins are multi-domain anyway, meaning that they consist of sort of multiple segments tied together. So adding another segment, adding another domain is not as such intrinsically problematic. It can give distortions, but nature has been using multi-domain proteins as a general solution for much of life's existence. And so adding another domain to something is by itself not fundamental grounds for concern. And indeed, people have made molecules where you have multiple fluorescent proteins embedded, and to a large extent, these work. Of course, there can be issues, but by and large, most proteins consist of multiple, or many proteins consist of multiple domains anyway. So fluorescent protein labeling is, to a large extent, a very well-working scheme. Much more challenging is labeling with synthetic dyes, and particularly getting targeted labeling. Of course, we would like to have control over what we label, and preferably even we may be interested in doing it in live cells, though we will talk more about this later. In general, of course, the scheme becomes, I have a target, I have a dye, I would like to connect them. And to do so, I'm going to rely on this magical X. Now, what could this magical X be? Well, that's going to be the focus of the next 10 to 15 slides or so, no more slides. And I'll try to give you an overview of the different strategies that are used. And actually, we should straight away distinguish cases where we do in vitro labeling, meaning that we have some purified molecule that we want to label in a solution, say a solution of some antibody or a similar molecule, and in cells labeling, where we have, of course, an environment that is orders of magnitude more complex, and where you can see that more subtle approaches are going to be needed. So let me just mention the very basics of in vitro staining. In vitro, meaning you assume that you have a purified system. Well, there you can typically use unspecific or less specific staining, meaning that you just make use of whatever chemical functionality naturally present in the target. And this can be typically, often you're looking at proteins. So the functionality that you use then are the functionalities present in the amino acids. For instance, the amine groups of lysines are often used. Diol groups of cysteines are often used. Carboxylic groups, the C terminus, etc., carboxylic amino acids or acidic amino acids. Essentially, within the natural repertoire of amino acids or whatever molecules, there are active groups or there are molecular groups that we can use to do labeling with specific types of molecules that will recognize those different features. And actually, 99% of these labeling experiments use just 2 or 3 techniques that have actually been around since the 1960s or even earlier. And a key target is often amine groups, which you have, for instance, in lysines, but also at the N terminus, you have an amine present. which you can label with different types of activated esters, such that in the end, you end up with your group, your fluorophore, directly attached to that amine group. A similar story also with isothiocyanate. You basically target any amines into the protein and you direct a dye to bind to that location. With thiol groups, typically cysteines, you use 99% of cases maleimide linkage, which again reacts and forms a stable covalent adduct that can have a dye of your choice. So in this way, you directly have covalent labeling of your molecules. If you buy labeling kits commercially, labeling with various types of molecules, be it dyes or be it other functionalities, by and large, what you will get is one of those. Either NHS esters to target amines or malamides to target thiols. And here something is wrong with the ordering. Actually, what I wanted to talk about first is what you have to keep in mind, of course, is that if you target amines, there may be many of those on the sample, and your labeling is likely to be very heterogeneous. So you will have molecules that are unlabeled, labeled once, multiple times, etc., simply because there is typically a richness in the number of fluorophores present in the sample. Cysteines, and this can also, by the way, interfere with its function. I will come back to that later. Cysteines are often used for site-specific labeling, but then such as for single molecule FRET. And in that case, typically you have to engineer cysteines in the location where you want them, and you may also have to suppress them elsewhere in the protein. So this works well as long as you are in vitro. What if you go into a more complex system, such as a cell? Well, in that case, you need to have some kind of specificity. You need to have some kind of molecular recognition. And if you're very lucky, you can use direct staining, meaning that you can use dyes that actually intrinsically, this is the dye moiety, a BODIP, that intrinsically have come with functionality that will achieve some targeting. Now, one example, for instance, are paclitaxel variants that immediately target the microtubuli in the cell. So this is really trouble-free labeling because you just get the dye, you add it. If you are lucky, it is even compatible with live cell staining. Otherwise, you have to fix and permeabilize the cell. But you have very trouble-free staining of the structure in question. Other example are the DNA dyes that we know, Hirsch dyes, for instance, that will recognize DNA. Different types of tracker dyes, mitotracker based on positively charged dyes that will go to mitochondria, ER tracker based on recognition of specific drug-like group derived from a pharmaceutical agent that will recognize microtubules. So this is overall very easy. There are always questions. Is it cell permeable? Is it specific? What is the effect of excess dye? If I have too much dye, what happens to it? Does it form a precipitate that can be problematic? Does it stain somewhere else, etc., etc.? And is it live cell compatible? So these are things that have to be evaluated. But if this exists, then this is a very easy and trouble-free solution. Of course, the majority of targets do not have such direct stains available. So what do you do then? And you can understand that this is a challenging question because what we're then really asking is, okay, how do we get a dye to recognize one of these molecules among this mixture that is present inside cells? Now with fluorescent proteins, we didn't really worry about it all that much because we could just do genetic fusions. So what are we going to do for organic dyes? Well, we're going to do basically the same thing. But instead of adding a fluorescent protein to the gene, we are going to add a tag, a stretch of amino acids that is recognized by the dye and some reactive group bound to it in such a way that we end up with a labeled protein at the end. So here what we are really getting at are so-called chemigenetic systems that are hybrids of a synthetic component, the dye that we have to add and that we have to get from a team of organic chemists, and a protein-based component that gives us our specificity, that gives us our staining. Things to keep in mind always, what is the labeling efficiency, what is the labeling specificity, what happens to unreacted dye, and these are always questions to think about as you are planning or doing an experiment. So one solution are the so-called self-labeling protein tags. And this was actually pioneered by work by Roger Tsien, and you will see Roger's name popping up quite a few times in this talk because of the many contributions that he has done to the field, beautiful contributions. And actually here, the idea was that, okay, if you can build in a selective chemical motif into a protein that can react with an organic molecule, then you can obtain a specificity. And in this case, the so-called FLASH system. The idea was to have 4 cysteine residues located in a particular configuration that would be able to react with this particular type of molecule. And this was a beautiful idea, of course, and it works very well because this is a motif that doesn't really occur a lot in nature. And so they could very nicely show that indeed you selectively stain those molecules. In practice, the system had some downsides. The staining specificity is not always high. The kinetics are slow, so it is not used all that much to my knowledge. But it was sort of a beautiful hint of things to come. And something that was especially beautiful about this system, and that I'll come back to later, is the fact that the product itself, the dye, is essentially non-fluorescent, but becomes fluorescent once it is added to or staining the target, which is fluorogenic labeling. And this is beautiful because it means you don't really have to care about unreacted molecules. So this was really pioneering the way. But I think now it is not used as much anymore. And probably the most used technique right now is enzymatic tag labeling, where actually we take our protein and we attach an enzyme that will recognize the label and that will bind covalently to it. And the example here are techniques such as SNAP labeling. So we have a protein to be labeled. We have a protein tag that is an enzyme. It reacts with a variant of the dye to give you a direct covalent bond. So this is a SNAP labeling. We have now halo labeling. We have clip labeling. And these, I think, are now the most widely used techniques to have dye labeling specifically in cells. So how do they work? Well, halo labeling, for instance, is based on a bacterial dehalogenase that naturally catalyzes this type of reaction. So it attacks halogenated carbons in order to remove that halogen atom and to replace it simply with an OH group. Now, of course, we can make analogs of such molecules, dyes containing such aliphatic chains and then a halogen atom. But of course, if we give that to the bacterial dehalogenase, then nothing much happens because it would just replace that with an OH group. So the key insight is really to mutate the enzyme, and this was the halo tag development, such that this reaction cannot occur anymore. And in that case, what you end up with is a direct covalent bond between the enzyme and the substrate, be it then the organic dye. So you have direct covalent labeling based on the fact that this reaction really takes place via this enzyme that you introduce into the cell. And overall, it works beautifully well. This is an example of work that we did where we have fluorescent protein labeling, but then also comparison here with halo staining, and you see that the staining is basically beautiful. So this is, I think, the main sort of challenge being done today. Because the binding is covalent, you can also do things such as pulse chase labeling. You can also do dual color imaging by having both SNAP and halo. But overall, these are really, I think, the most easily applicable strategies that we have now. And much development is being done on optimizing these systems even further. What is a third approach? Actually using protein tags, small stretches of amino acids that are recognized by another enzyme that you have to introduce into yourself, such as a sortase. And that will recognize, again, your labeled dye, be it a probe that has an oligoglycine and that has a sequence of glycine residues. And this enzyme will recognize this label, it will recognize the tag, and it will fuse them together. So an example here is sortase labeling. In fact, there are other types of labeling known as well. To have a more better view, I would like to invite you to simply go and look at this review because it goes into a level of detail that I can't really do here. But the nice thing about this is that you end up with a system where you only need a small protein tag. You don't need to fuse an entire enzyme, you just need a small protein tag. And you can have direct covalent coupling. The downside is, of course, that you need to have this machinery present. You need to have that enzyme present, and the labeling itself can be slow and need a very high concentration. So while in theory this can be done inside live cells, often I see this done in vitro in the majority of cases, but there it can be absolutely beautiful. For instance, this is a study where they developed a nanobody, and they started by staining this body, a nanobody with the classical NHS staining. And what you see is that you have a fairly poor staining of your cells. However, if you compare it with staining via sortase expression or the sortase system, you see that you have much more better staining because you have a fairly localized tag, a short tag, where there is just one copy at a well-known location, and so you end up with one label, as opposed to the more stochastic labeling intrinsic to NHS coupling. Of course, this system can be generalized to any other type of molecular functionality. So a whole range of different molecules have been designed to do this. So this is indeed a beautiful system. Other such systems exist as well. This is a system known as SpyCatcher. Again, here you have 13 amino acids that you have to fuse onto your target. So it's a small peptide stretch. You can have it on either terminus or whatever. And then you have a second element, protein, SpyCatcher, that will bind this covalently. So if you attach your dye here, you will again have direct labeling with many of the benefits as you have with sortase-based labeling. But again, mainly, I think, easy to use if you have systems that you can engineer or in vitro, because you do have to introduce this protein, which is hard to do to get it to go spontaneously across cell membranes. Another solution, non-canonical amino acids. This is very nice in concept. The idea is that you replace part of the protein dictionary, the amino acid dictionary of cells, with a codon that will give incorporation of a non-natural amino acid that will then have some particular functionality that can be recognized. But in fact, this can work beautifully because this is about the smallest tag that you can have. The downside is typically what makes it challenging is that you have to engineer the molecular machinery of the cell. You have to have new transfer RNAs, you have to have new enzymes that load those transfer RNAs. And it's hard to do this for organisms in general. And second of all, it may also be tricky to do this for more than one label because you need to have room within the genetic code. So this is a system that is extremely nice, but more challenging to apply in practice. And then maybe the final solution, or close to the final solution, because before we do some kind of questions, but a family of labels that has been making a lot of inroads in the recent years are so-called fluorogen binding or fluorogen activating peptides. These are basically proteins, so protein components that you can fuse to your target in the same way as a fluorescent protein. But the difference with a fluorescent protein is that they don't make a chromophore automatically, but instead they bind another molecule that you have to supply in solution. So you have cells expressing a protein tagged with such a peptide, and you have to supply an additional molecule that needs to be cell permeable. And when it is bound by this protein, you end up with a fluorescent group. Now, this is reversible, which means that the dye binds and unbinds spontaneously. So your free cofactor, this molecule, needs to be present in medium at all times. And the system also has to be fluorogenic, meaning that it can only become fluorescent. The molecule needs to be dark until it is bound by this protein. But this can be used to get beautiful staining that is wash-free because of the fluorogenetic nature. And a range of such systems exist as well. This is another system that actually uses biliverdine as the cofactor. And as you can see, both components are non-fluorescent, the protein component and the cofactor, until they mix and then they become fluorogenic. The fascination here with this system is first of all the genetically encoded nature, but second of all also the reversibility, which I will come back to later. Other systems exist as well. These are not the only ones, but I'll get back to this in a moment later as well. Other examples include the so-called infrared fluorescent proteins. These molecules actually also live in such a scheme where they bind another molecule that then becomes the chromophore. Has seen a lot of attention as well, and the goal here really is to go into far-red imaging with such dyes. But if you can't or don't want to modify the molecule, or it's not a protein, well, then you should use something that can bind it. For instance, a nanobody that can bind it, that is then fluorescently labeled. This can give you problems because it's always fluorescent, also when it is not bound. So there exist schemes to try to make this solve this issue by having the non-bound nanobodies be degraded. There's also lots of other schemes that can be used, but basically in that case you simply need to have something that is conceptually similar to an antibody that will bind it. Voila, so this is an overview of some common strategies that are used. Maybe one last thing that I want to highlight is that these molecules all have certain sizes that can interfere with the function of the molecule. This is an organic dye. This is fast, nanobody, SNAP, GFP, HALO, an average-sized human protein is about 56 kilodaltons. So you see that the sizes of these labels are non-negligible. On the other hand, you have to remember that the cell is a very crowded place anyway. So the problems of having such labels are not always as strong as they may appear. Okay, some thoughts on live cell labeling, and now especially for you, Vanessa, because this answers your question. First of all, my thinking, if you can have a well-working dye stain, something like DAPI trackers, etc., the first preference should probably be to use those. If you can get them into the system in a way that works for you, that's probably the easiest solution. Otherwise, if a fluorescent protein meets your needs, then use that. Then don't go into other systems to begin with. If a fluorescent protein is okay, just use that and life will be okay. Consider synthetic dyes if you need high brightness, high photostability, tunability, etc. Or perhaps last-minute decisions on what you want to use. If you want to be able to change the wavelength as needed of your dye, then consider using synthetic dyes with one of the systems that we had before. In general, now I see the strongest push for HALO and SNAP. We find, and others as well, that HALO is a better working system of the two. So start with that and then consider SNAP if you need it. Fluorogen activating peptides, something like FAST and the like, you don't need to form a chromophore. This can be work in situ, in hypoxic conditions. You can have bigger spectral reach. And again, you can have postponing of the choice of label. You also have the advantage and disadvantage of reversible binding. I will get back to that later. One thing that we have found is that background from unbound fluorogen can be a concern, and actually getting the fluorogen in there can in some cases also be a concern. More specialized types of fluorophores that I haven't really talked about, quantum dots, nanodiamonds, and the like, are mainly interesting for use cases where you really need their optical feature. Really need it. Things like long-term single particle tracking of a protein on a membrane, but labeling can be tricky and not something that I'm really discussing here in this talk. Okay, so this is this part on live cell labeling. So maybe Gertrude, if there are any questions, we can take a look now.
Yeah, can you hear me, Peter?
Yes.
Good, because yes, thank you so much, Laura. I also hope that your question was answered. If you have any additional questions, please put them on the chat. In the meantime, there are people already posted chats and questions. The first question comes from Vladimir. He says, introduction of a plasmid is associated with secondary expression of a protein of interest. What expression control tools are available and how hard is it to implement them? Thank you.
Yes. So first of all, this is a question that really ends up being a topic in terms of genomic manipulation of the cells, right? So it's not really about the label chemistry. First of all, the easiest solution is typically to use plasmids. Plasmids, indeed, the expression level might be different than the original target or than the native target. First of all, you can try to adapt to this by selecting promoters in different ways. So that is an option that exists. You can try to have the same promoter, etc., etc. But even then, this is not guaranteed. So you can try to approximate, but it's not guaranteed. And you also end up with the fact that you have now additional copies of your molecule. So you have your native protein, and then you have your native protein plus one that is labeled, plus the label. So this is essentially a problem that simply is there and that is difficult to remove in such a way. The next level up is to have stable cell lines. These essentially avoid features associated with the variability of plasmids. So you don't need to transfect your cells because it is stably integrated into the genome. However, typically the integration is in some random location, and the expression level will again be different compared to your native protein, and you will still have the problem of having a mixture of native and labeled protein present. The next step is also you can use cells where the native protein has been blocked, knockout lines, and then you add a plasmid or a stable cell line that express your labeled molecule. Then you get rid of the presence of two populations, but again, you're not fully sure if the level of expression is the same. So then the next step up, if you really want to do this, then you want to do something like a CRISPR knock-in, meaning that you actually genomically replace your protein with a gene that has your protein plus the label. And this is, of course, the best solution in terms of having just one population at the right expression level. But of course, it's also the most amount of work. It may also still cause a bit of artifacts, but it is the closest way of getting to the native system. It is still a considerable effort, but okay, with the pace in genome engineering, this is soon going to be, I think, something that is very manageable. And even now you can get this done as a service.
Okay, next question is from Sujoban. Regarding the flash labeling, why is it not used so much? Has this to do something to do with toxicity in cells?
Yes, indeed, it is not used as much. I think that key reasons are that there's actually a significant amount of background staining, at least from what we have observed and also what I see reported in literature. The molecule reacts with other features in the cell, meaning that you get basically background staining. The kinetics of the labeling can also be tricky, and I think there are also inherent toxicity considerations in working with such compounds. I think it's a beautiful system, but today what I see is that it is mainly interested as sort of having really defined this field, but it is not used all that much, by which I don't want to mean that it is never used, but I think it has not reached the amount of applications that you see now with Halo and SNAP domains and the like.
Okay, thank you. Then we have a question from Hani Esmaili. Regarding different ways of labeling, do the different methods affect on spectrum of fluorophore, and what about degree of labeling?
Yes. Do they reflect on the spectrum of the fluorophore? Yes and no is the answer. It depends a bit. First of all, fluorophores typically, so we're talking synthetic dyes, I guess. With fluorescent proteins, typically we assume that there is little effect. If we talk about organic dyes, then first of all, there's a couple of things that change. Typically, you need to make variants of these dyes. You need to have this halogen alkane chain or whatever. So you need to synthesize variants of the dyes. This may already induce some kind of change, though typically the functionality is away from the conjugated system, so you can expect this change to be small, but we cannot exclude it. But then, of course, once a dye is labeled, your environment can change. So it can go from a polar aqueous environment into an apolar environment, and then you can have starting changes, get changes in emission spectrum. So the answer is yes. It can affect the features. It can affect it because you need to change the structure. It can affect it because you change the environment. And actually, this is a big feature, and I will come back to this later. But in practice, this is not really something that you're likely to encounter, or let me rephrase, in practice, this is something that needs to be taken into account from the beginning of the measurement, and really depends on the dye that you use. But I think it is unlikely that you will run into a situation where you use a dye that has been validated for halo tagging, and all of a sudden you have a completely different spectrum. But yes, of course, compared to the native unmodified fluorophore, there can be changes in the optical properties.
Then I have a question from Valeria Berno. I'm not sure if you're going to cover this later on, but she's wondering if you can speak about dyes and mounting media in fixed samples.
Ah, right. Yes. I'm actually not going to cover that because I have assigned myself a bit of a focus on live cell imaging. Yes, mounting media can have a strong effect. However, I should make a disclaimer that I'm not really an expert on mounting media at all. So no, it's not going to be covered. If you have specific questions, then feel free to ask, but it's not going to come up later in this talk.
Valeria, I also think if you have specific questions, maybe you can forward them, Peter. I also know, I must say that I think this is a very important question though, because I know that there are mounting media, you have different types of mounting media, which is watery based or more resin based. Depends also the mounting media that you want to use, depends on the type of techniques and microscopy techniques that you use later, for example, STED or a particular refractive index. And there are dyes, I think, well, I noticed that certain mounting media, and where's the critical part in mounting media, is that they sometimes include anti-bleach agents. And for example, the Psi dyes, from my experience, cannot handle anti-bleach agents because there is no fluorescence anymore. So in these particular mounting, let's say if you use cyanine dyes, I can recommend to leave out any anti-bleach. And for example, perhaps you saw some of my papers also, I do a lot of thread experiments. There we can never use anti-bleach, any mounting media with anti-bleach reagents in there. So I think, Valeria, this is indeed a very important question. I also think that unfortunately might not be the right, because it's an extensive, this can really go into a long discussion. I'm happy to chat with you also and with Peter if you have specific questions to that. So you can, we have all of us, we have my own chats. So please send us a chat with specific questions if you have to, and we are happy to address them. So then, Peter, I have Grace. I'm sorry for maybe my pronunciation. Grasvidas. He's asking, or she's asking, do you think that currently it is enough variety of dyes and tags available? And maybe you can even talk about your own future perspectives on this.
Yes, and this is something that will actually come back also to some degree later in the talk. I think the answer is right now for fluorescent proteins. There, the main challenge is having the variety of labels as such. Labeling, how to label with them is not really something I would say that is the main challenge. The challenge with fluorescent proteins really is having a big enough spectral range. And of course, it's never big enough. And people are really working on increasing that multiplexing, and I'll come back to that later. I think in terms of chemi-genetic systems, this is still a major challenge today. For instance, what you typically hear is people use halo, and then they use SNAP, and then you have two ways of staining, right? And then getting a third way of staining, okay, you can use, you can get lucky and use something like a tracker dye, but having really three fully genetically encoded ways of staining, for instance, is to some degree a bit of an issue. And I think that this is one of the key challenges that we face today, not only with such systems, but also, for instance, with the fluorogen binding peptide conditions as well. So this is a major challenge. It's something that people are really working on. But the answer is that, yes, I think that nowadays that is a strong limitation that urgently needs attention. But I'll get back to that a bit later on.
Okay, so so far for the questions, I would say that you still have next chapter, I think, of your talk.
Yes, one or two, two upcoming chapters, more or less.
Okay,
well,
yeah,
three.
because you might be surprised, Peter, it's already 11:30, and I think it doesn't matter. We continue like you wish.
Yes.
But let's continue, and I'm looking forward to the next chapter or session.
Yes, thank you. Time is flying indeed, but I very much appreciate the questions.
Me too.
Nice to have so much discussion. Thank you all.
Yeah, I also thank you all for your participation, and let's continue. And looking forward to the next questions from.
And here is the next chapter. So now I'd like to also shift gears a bit to a topic that is really close to my heart, which is what I call smart fluorophores. This is why in the title I wrote combining the beauty of chemistry and physics. First of all, I didn't really talk about physics, but I think it's evident that, of course, the instruments that we use, the devices that we have to do our measurements are really based on the laws of physics. And so I think the beauty there is evident in the way in which these systems operate. But what I'd like to talk about is really the combination with the beauty of chemistry. And we've seen that to a large degree already. Yeah? We've been talking mainly chemistry for all of the talk until now. But I'm also interested, or very much interested, into something that goes another step beyond this, and what I call smart fluorophores. And let me give you some examples of what I mean by smart labels, which are really fluorophores that play an active role in the measurement logic, meaning that they do more than just emitting photons. And we know those for quite a while now. This is, for instance, two examples where here we have a green-red convertible fluorescent protein. So this becomes red when illuminated. This is a photochromic label that can be switched on and off. And these movies are actually coming from the groups, the Miyawaki group in Japan, who was one of the first groups to discover these types of behavior. But what you see here is that really these molecules have a logic to them that you can control and that you can really take advantage of in the measurement. These are light sensitive smart labels to me. Of course, the threshold for being smart is lower. Anything that has a more active role. But in the logic itself qualifies as a smart label. And another example of a smart label are actually biosensors, environmentally sensitive molecules. And here, for instance, we see a calcium sensor showing us waves of calcium signaling within a developing zebrafish. So these are truly fantastic molecules, and they really become part of the measurement logic. And so they are as integral, as important for the proper functioning as your experiments, as are the various devices that you have here. And they really contribute to the measurement itself beyond just emitting photons. So of course, these are now known for a while now. We know that there are different types of light-induced smart behaviors, and these are the keys to doing, for instance, different types of subdiffraction imaging. So really, these molecules are part of the measurements in a way that goes much deeper than just emitting photons. So how do they work? Well, of course, it's going to be beautiful chemistry again. This is an example of a photochromic fluorescent protein. So this we can switch on and off with light very beautifully. How does this work? Well, there is a change in absorption spectrum when you illuminate it with light, which is due to a protonation of the chromophore. So if you have a protonation here or a deprotonation, you completely change the emission spectrum. Then we also know that there is a cis-trans isomerization involved, a photochemical transformation. And so overall, you have a system that has 4 molecular states where only one of them is fluorescent. And really performing the measurement requires that you transition the molecule between those different states in much the same way as in a typical measurement you would transition your excitation light sources, your detector, your filters between different states. So they really are part of the measurement, but it is embedded in the chemistry. And this for me really is the beauty of the chemistry involved. This is often an intrinsic property of the molecule. For instance, we modeled this photochromism recently across a variety of different FPs or different mutants, and we find that just one mutation is enough to completely change the spectral properties. And what this tells us is that these systems often make use of the fact that the fluorescence really depends very strongly on the molecular environment. So small changes in structure have big impacts on the fluorescence emission, and that is something that we will come back to later. Again, fluorescent proteins, photochemistry can be complex and involved. It can also involve interactions with the environment. This is a paper from a while ago, but we actually showed that M-cherry could become photochromic if you add a molecule called BME. And so you could take these samples of M-cherry and switch them on and off with light, and then you could use this to do single molecule localization microscopy with a nice result using just M-cherry. But we actually looked into this in detail, and we found that you had a mixture of events that caused this, including direct addition of your chromophore. So really, smart behaviors can arise in a number of different ways, but by and large, they always have to do with the chemistry of the sample and the degree to which you can apply this to your ends. Other examples are green to red convertible dyes again, where of course you can do things such as single particle tracking. So I'm not going to go too much into detail on this. This is a nice overview of how these mechanisms actually work. But the point is that really here we are bridging the gap between traditional imaging, where you see the molecule as just a source of passive photons, and schemes where really the molecule becomes an active partner in the experiment as well. So this, as we now know, this also exists for organic dyes. Here we have systems such as the well-known D-storm system, where you have reagent-induced switching of molecules between different states, on and off states, that you can then use to do super resolution imaging. Other systems exist as well, all really with the goal of manipulating the chromophore chemistry in a way that we can take advantages of it to boost our imaging. Now, just to give you an idea, here we have two types of smart labels. We have a fluorescent protein, green to red convertible. Here we have a photoactivatable JF dye. This is a study that we published 3 years ago now. And what you can see here is, again, with these photoactivatable dyes, you see the performance that you get out of organic dyes compared to fluorescent proteins. But there's a caveat as well. This is halo-based labeling, and there you see that this photoactivatable dye actually had a lot more background staining. So you always have to weigh a bit the pros and cons of, okay, what does a particular system do for me? But overall, a broad range of such schemes exist. So we can have photoactivatable organic dyes, photochromic dyes. We can have different types of light-induced switching, and also something that I'm not really going to talk about in this talk that much, but now very cool as well, are these spontaneously blinking dyes. So these really allow you to lift your imaging to the next level. In addition to things like super resolution imaging, what you can do is things like dynamic labeling. Meaning that you use one of these dyes that you can switch on and off photochromic dyes to see, for instance, shuttling between the cytosol and the nucleus and the other way around, which helps because otherwise, if you have a steady state distribution, it's hard to really know what goes on. In this stage, what you end up with is something like dynamic fluorescence recovery after photobleaching, FRAP measurements. Also, photoactivation can be used in this way, basically allowing you to see the fate of what happens between molecules. Now, while such smart dyes are cool and they really add a whole new dimension to probe engineering, there is also such a thing as dyes that are too smart. And examples here include, for instance, photoconversion of dyes between different states, such as these dyes that are often used for single molecule imaging, etc., where it was shown that they can actually convert into species that have different emission spectra, and that this can actually lead to artifacts. And we have also seen an example of that already, of course, in the sense that you could say that photo destruction is already also smart behavior. Yes, but in this case, also a bit too smart for most of us. One behavior that we actually have seen a couple of times before is this fluorogenicity. Fluorogenicity meaning I have a label, and when it targets, when it reaches that what it is supposed to label, it becomes fluorescent, and otherwise it's dark. And this is really a really crucial element that people are now heavily pursuing, because of course, if you can avoid your dye from being bright when it is not bound, then you can eliminate a lot of washing steps that you would otherwise have to do, and you may also eliminate a lot of background signal that you would otherwise have. So fluorogenicity is something that is really seeing a lot of attention nowadays. And just as an example, remember the FAST system, where we have a protein that binds a molecule, and when it binds this, it becomes fluorescent. So how does this fluorogenicity work? First of all, again, here's the illustration. When you don't have the protein, the dye is dark. When you have the protein, the dye is bright. So how does it work? Well, typically it relies on two effects. On the one hand, shifting the absorption spectrum can allow you to get fluorogenic behavior. And on the other hand, having something where the fluorescence quantum yield is higher, where it is bound, can also give you fluorogenic behavior. And this molecule really does both. Because when it is bound, the pKa of the molecule changes. So you lose a proton here. And losing this proton actually gives you a shift in absorption spectrum. And what this tells us is that if you can have your labeling interfere with your conjugated system, your system of delocalized electrons, you can have a shift in absorption band, which would manifest as fluorogenicity. But second of all, this molecule is also non-fluorescent in solution, because when it is excited, it undergoes fast cis-trans isomerization, meaning that it is non-fluorescent in this state, but then when it is bound, it becomes more rigid, and that rigidity allows it to be fluorescent. And actually, getting more rigidity is a key element driving why something is fluorescent. Rigid molecules tend to be more fluorescent because they have a harder time losing their energy as heat. Other mechanisms exist as well. For instance, the di-yo-yo-1 is fluorogenic because it forms a non-fluorescent aggregate in solution, but when it binds to the Na, this aggregate is broken. Other examples that can be used is, for instance, dyes, here derivatives of the silicon rhodamines that can exist in two states in an equilibrium, one of which is fluorescent, one of which is non-fluorescent. And it turns out that this equilibrium can be controlled by binding, in the sense that, okay, when it is bound to the protein, it will exist in this state, but when it is non-bound, it will exist mainly in this state. So those are a couple of ways in which we try to obtain fluorogenic behavior. And this is really something that is strongly being pushed now because it makes life much more convenient. Overall, just to summarize what you have to do to get fluorogenic behavior, you change the fluorescence quantum yield. You rigidify it, for instance. You change the absorption spectra, either by covalently modifying it or changing some molecular equilibrium upon binding. These are, of course, ideally combined. Something else that I want to briefly touch on are chemically smart labels or biosensors. Maybe I'll continue with this part and then we can ask questions about all of the smart label business. And these are environmentally sensitive probes, and they're really cool molecules. I have shown the calcium imaging in zebrafish. The question to ask, this is a zebrafish embryo that's beginning to divide. And you see these beautiful organized, synchronized calcium events at these first divisions, which I think is absolutely fantastic. And we can ask ourselves how we could see this without having molecules such as biosensors. This is a cell division, also in embryonal development. These are oscillatory signaling processes within cells expressing kinase reporters. And these are molecules that are sensors that indicate different states of cell death. So these are fantastic molecules. And how they work is really by having a fluorescent that depends on some aspect of the environment. Now, making biosensors can seem easy, which is great news. Why? Because let's say you have a probe that has a perhaps troublesome environmental dependency. Well, then you might really have a glass half full, glass half empty moment, because what you could do is call it a biosensor, and then your dependency all of a sudden has become a feature. So this is something that actually is a pretty neat way at looking at things. And it is also used in practice. For instance, green fluorescent proteins have a significant pH dependence, depending a bit on the mutant that you have, meaning that the fluorescence strongly depends on pH, which means that indeed what you can do is take this pH dependence and then you can see it as an issue, or you can actually turn it into a pH sensor. And this is one of the first papers that actually did this, and it's a beautiful piece of work. It's not I'm not saying that this was easy work or whatever, but the fascinating thing is that they just use an intrinsic dependency of the probe. So if you have such dependencies, you might see it as a problem, or you might actually see it as a biosensor. Actually, here I couldn't choose between can or is hard, but making biosensors can be very hard from an engineering viewpoint, from a chemistry viewpoint, because if there is no suitable dependency between your dye and the target that you want to see, then you have to add one, which means you have to find a molecular component that has one, and then you have to tightly couple it to the properties of the fluorophore. And that is really where the difficulty comes from. If there is no natural dependency, then you have to find some molecule that will react to your component and translate that into a change of fluorescence. And for instance, within genetically encoded biosensors, there are a number of schemes that have emerged. And maybe I'll just give you a few examples of that because it's very neat. One example makes use of sensors based on just a fluorescent protein that has been designed in such a way that when you add two interaction partners and they interact, you end up with a fluorescent molecule. And the prototypical example there really is calcium sensing. And how they really work is they use a variant of a fluorescent protein that has a particular insertion, or a protein where you have a particular insertion that was actually identified by accident in the 1990s. And that can affect this equilibrium that we had between the chromophore being protonated and deprotonated. So it can affect this equilibrium between the dark and a dim state, a dark and a fluorescent state. And indeed, what you see is that when binding happens, what you really do is you involve this protonation state. And so you can have fluorescence that is controlled by whether these two moieties interact. And this is an example where such molecules are used in neuronal imaging. So here you see neurons in a brain slice, and you see them lighting up when there is neuronal activity by coupling really the chemistry of the chromophore to these two associating molecules. This concept can be generalized. This is an example where they did it to make a sensor for hydrogen peroxide. What do you need? Again, some kind of peptide motifs that will show association in the presence of the stimulus. And that then really becomes the element to consider. This is another example, seeing hydrogen peroxide in action using such a molecule. Another design is based on FRET, and there the prototypical example are sensors for calcium, where you have binding of two peptide moieties in the presence of calcium, and you end up with a change in FRET that you can then visualize optically. Of course, nowadays these systems have been optimized since this pioneering development, and the example here is again this zebrafish embryo. But the system is general, and many sensors have been designed in this way. Other things are insertion of a fluorescent protein, and this is an example where they use GPCRs. And actually, sorry, I wanted to show this first, which are very important membrane receptors, and it is known that there is a small conformational change or a conformational change upon binding. Well, by inserting a fluorescent protein at the right spot, it turns out that you can get that conformational change to affect your fluorescence. And so you end up with a sensor. And the really nice thing about this is that it makes use of the fact that fluorescence is very sensitive to its environment. And so even a small change can translate into a big fluorescence contrast. Biosensors are really a broad field. I can't really do it justice here, or at least do more than scrape the surface. So there are some very good reviews out there. I just want to highlight also that with organic dyes, several biosensors are known, and indeed originally organic dyes were the tools to use, of course, because we didn't know about fluorescent proteins. And maybe very quickly, a couple of examples just to illustrate that it is really the chemistry that enables this. One example is the use of dyes that probe membrane and other viscosity environments. And these have to do with molecular rotors that are really dyes that can undergo a fast, say, isomerization, a rotation of this moiety here, such that when you excite them, they lose their energy via that rotation and they are basically non-fluorescent. However, the trick is that this rotation depends upon the insertion into the membrane. If they go into the membrane via this alkyl chain, then the rotation is slowed down. And the degree of the slowing, and hence the degree of the fluorescence that is seen, depends on the viscosity of that membrane. So this you see here, for instance, where in this state, measuring with the lifetime, you really probe the degree to which this can rotate and hence the degree of the local viscosity. So again, this shows us that you can have molecules where you have some intrinsic dependence that you convert into a biosensor. Another example are solvatochromic dyes, where you have dyes, for instance, push-pull chromophores, that when they are excited, they become charged in a way you get a strong charge distribution. Now, this charge distribution, if you have a highly polar excited state, then in a polar environment, this state will be more stable. So this will go down, and you will have a red shifting of the emission. And in a less polar environment, it will go up, and you will have a blue shifting. And indeed, depending on the polarity of the environment, you see different emission spectra. And this then you can do, for instance, to probe the degree of lipid ordered and lipid disordered reactions in membranes. So you can start mapping those out. But again, what you're using is you're really taking those basic chemical dependencies and moving those into a optical readout for sensing. All right, so that's maybe the end of this particular part. So I don't know if there are any questions in the meanwhile about the past chapter on smart molecules.
Yes, Peter, there are several questions and I will start reading them loud. And the first question comes from Grasvidas. Do you think that do you think that fluorogenicity is really essential for obtaining a good image? What about the importance of specificity and affinity?
Yes, that's a very good question. Is it truly essential? No, it's not truly essential, but it can make life easier. It also depends a bit on the system that you have, but let's break it down. Because we're actually talking about a bunch of different concepts here. First of all, the question of specificity and affinity, meaning finding the right target and finding it with high affinity is of course essential. Whatever you do, that's a priority number one. Otherwise, you just get background staining or you might get weak staining. So if you have to sacrifice this in order to get fluorogenicity, I would say stick with the high affinity and the high selectivity. But really, the question that it always boils down to some extent, is what do you do with unreacted dye? If you have a perfectly specific system, then your staining is fine. But then if you have a lot of unreacted dye simply hanging around, then either you get bad images or you have to wash them out. And that washing out can be tricky, or it can be perturbing to the cells. So really, the fluorogenicity allows you to avoid much of that washing behavior. And it may give you a bit of an edge up in terms of getting that specificity, in the sense that, okay, even if you have some side affinity for something else, but if that something else maybe also attracts your dye, but then if that dye is less fluorescent at that location, because it is fluorogenic, then that may actually still be a win. So the question is a good one, but it actually contains a number of different elements. But overall, of course, having a high specificity and a high affinity is paramount. But then questions emerge into, okay, what if that is not achievable? Can fluorogenicity help to push us beyond that? And the second element then being, what do you do with excess dye? Can you get rid of it easily or not? But your question is correct. It is not a hardcore requirement, except for the fluorogen activating peptides that we've talked about. There it is essential because the fluorogen has to be available at all times because the interaction is reversible. And so if you have reversible interactions, that means your dye has to be present at all times. Otherwise, you just wash it out because it's not covalently labeled. Well, in such systems, you do need to have fluorogenicity.
Okay, thank you. Yeah, I'm looking for my microphone switch. So the next question comes from Krista Walter. Photo switching of fluorescent proteins is often done with UV excitation. In your experience, how much does this influence live cell imaging over longer periods of time?
Yeah. So my experience is, okay, we haven't really quantified this strongly, I should add. But one thing that I think is important to realize now is that in the on-off photo switching, so I should divide a bit. If you are talking about on-off photo switching, and maybe I'll just go back a bit to explain what I mean. If we are talking about this type of switching, photochromism, reversible, then one thing to realize is that off switching is, for most of those labels, the off switching needs much more light than the on switching. So the on switching is typically super efficient. Quantum yields are like 0.5, extinction coefficients are like 50,000 or so. So you actually need very little light to switch them on again. So my experience here is that, okay, it may have an effect, but this effect is small because we are talking small amounts of light, because this is an incredibly efficient process. If we are talking photoconversion or photoactivation, there it can be problematic because you need more irradiation. It's a less efficient process. It also requires much more chemistry, in a sense, to happen within the protein. And there are indeed studies that look at this in terms of phototoxicity, and they do find consequences of this. So I would like to invite you to just look those up to get the hard numbers. People have been trying to mitigate this via methods such as primed conversion, meaning you add a second wavelength that can make the conversion itself more efficient, or even triggered via different wavelengths. But yes, here this can be an issue. Here, my feeling is, I'm not saying that it can be an issue, but it is much less likely to be an issue because this is an extremely efficient process.
Okay, thank you. Then a question from Ilya Stoyev. Her question starts like this. My question relates to the use of fluorescent traces for extracting the mechanics of complex fluids or cells. Would you know of any tests or controls that one can run to probe for potential interference between the fluorescence tag and the process or structure that one is interested in elucidating?
Yes, that's a very good question. And that's a question that many of us have at all times, especially people who are applying the tools to investigate a complex mechanism. I'm afraid that there isn't really or at least as far as I know, there isn't really a magic solution to this. Typically, controls need to be done by looking at other parts of the system, in the sense that you try to look, for instance, with a number of different labels if you see a difference. And so one thing that you can do is you adopt a number of different fluorescent proteins, you adopt a number of different labeling styles, labeling strategies, and you see if this has an effect, if you see differences between them. That doesn't guarantee you that any labeling will not produce an interference, but it can allow you to compare if some techniques have more interference than others. Other than that, the easiest thing is to, or not the easiest thing, but another way is to look at ways of evaluating this that don't require the fluorescence. For instance, with the cytoskeleton being helical or not, this was clarified by having a look at electron microscopy images. Now, I'm not saying that you should do electron microscopy, but if you have some kind of functional readout or some kind of assessment process that is independent of the labeling, even if the cell is fixed there, the cell doesn't have to be alive, the system doesn't have to be alive, but if you can have any ability to see changes there, then that will help also. But I'm not really aware of a really magic bullet to this. I don't know, maybe Gertrude, you have some thoughts? Because it is a big question, but a tricky one as well.
Yes, I'm also thinking it is a tricky question. I think it's a lot of controls indeed, a lot. So it's not one particular control. And I also see that, let's say, I try to overlook the interferences in the processes. For example, when doing FRED, and I try to see if, let's say, if it's going between labels or can change. And maybe it's the best to see if you can reach a structure readout process in with two different readouts or two different processes. That's the only thing I can say at the moment. But I think this question also here, Ilya, if you have a very, maybe you are having something in mind because of your own research, and then it can be a bit more specific. So if you wish, please contact us and on our own private chats, and it might be easier for us to address in a more specific way, because this is quite broad and alone. Also, Krista, thanks you for your answer. So, Peter, I know that you're flying blind, and I just want to let you know that we have around 10 to 15 minutes left for you. If I think you still have a chapter more.
But it's a fast one. Okay. I think so.
I just wanted to let you know, and if you had let's say 2 chapters or so, I would have liked to ask you to choose one of them, but then I would say let's continue. And thank you so far, and all people, thank you for your contributions, because I really like the lively discussion that we are having now.
Yes, and indeed, I fully agree. I'm enjoying the discussion. I was a bit worried about that with the online format, but okay, it's been working out well, so thanks. I'm just going to get back to where we were at, and then I'll continue. And actually, I've sort of covered a bit the background that I wanted to do. The field is so broad and so diverse that really I had to be very selective in what I picked. Sorry for that, but okay. That's how it is, given time limitations. What I would like to talk about a bit in the last chapter is simply some trends that I see coming up, some areas that I think are seeing work, and I'm really very selective here. So really, I'm missing out on a lot of exciting work, but just a few key points that came to mind among many other possibilities as well. So first of all, people continue to develop improved labels, including, yes, smart labels. Sure, I love them, but also the more classical labels. Improvements continue to be made. And this is, I think, something that is important to realize, and also in a way fascinating, because we keep, even though overall the set of basic dyes that we know is limited, the number of scaffolds that we know is limited, but we still manage to keep on tweaking those to get significantly better probes. And so some highlights are indeed things like fluorescent proteins with extended brightnesses, extended spectral ranges. So those keep being developed, keep being pushed. Even surprising developments in a way. This is a new green fluorescent protein. So green fluorescent proteins have been investigated for a long time. But even there, surprisingly, we still managed to find new labels, such as this M-green lantern, where if you compare it with other dyes in cells, they find a really strong improvement. So there is still continuing gains being made. So don't remain limited to whatever you used during the PhD, etc. No, things do keep on improving. Also within smart labels, photochromic labels, process continues, progress continues, new types of screening being used. new types of photochromic smart labels being invented, even mainstays, things like cherry, seeing significant improvements and the like. Also organic dyes, I'll get back to those later or in a bit as well. But essentially, what is there is still room to be had in improving dyes, significant space to be had in improving dyes. Also, new types of labeling. This is a nice study. There are others like it. So this is not the only study by far, but also approaches of designing labeling schemes to do things like protein paint, super resolution imaging. Also, again, in organic dyes, this is a figure that I showed before, where actually, photon budgets are considerably increased by rigidifying the structure and looking at the substituents. So even there, improvements continue to be made. New dyes emerging on the market. Of course, we know this. We've seen, for instance, the JF dyes coming up and taking over really a lot of space, not only because of their good performance, but also the fact that financially they were very interesting in terms of purchase price, being distributed at very low cost by Generia. This ties back to a question that we had before, where there are ongoing pushes to extend the spectrum. And actually, these are systems that really go into shortwave infrared, try to push beyond 1,000 nanometers, for instance. And what you see in these molecules are very long conjugated systems. So you see that these are going to be molecules that may have some tricky properties to be used without going into too much detail. And typically also brightnesses are low. This is what I mentioned, low brightness, specialized equipment to some degree. And so the types of images that you get out are very different compared to just cells, where we see subcellular structuring. These are more like whole body imaging and the like, partially also because the brightness of this system is so low. But nevertheless, this is a fascinating area where, again, we keep pushing dyes forward. Other dyes for super resolution microscopy continue to be made. Here, this is a nice example that I thought, because I like smart dyes, but here actually we are making dyes less smart, in the sense that this is a dye that is designed to not have this photo bluing effect as well being illuminated. So this is another fascinating development. So new dyes continue to be made, which of course begs the question of how do I know which dyes to pick for my experiment. And I think the answer there is we've seen a couple of databases coming up. We have FP-base for fluorescent proteins. We have databases for sensors. For organic dyes, I think I'm not really aware of a formal effort there. Maybe I've overlooked it. That's possible. But I think very often the key is in talking to users, talking to the developers, asking which dyes work well, etc. But there are much better labels out there compared to what we had X number of years ago. Another push that has seen strong efforts is really lifestyle compatible imaging, mainly then in terms of organic dyes, because for fluorescent proteins, this is a given. But a lot of push towards fluorogenic probes for easy labeling now, a lot of it based on Halo, SNAP, and related approaches. This is really something where a lot of effort is being spent on trying to have good fluorogenic, highly selective solutions that can be used and also for things like super resolution imaging. Here another one, fluorogenic dyes for easy use, similar approaches here. Really a lot being pushed now towards live cell imaging, and preferably experiments where you just add the dye and you don't even need to wash it to that extent. Of course, in practice, there is still a middle ground. We have found that it can be tricky. To stain with different fluorophores, you're likely to need to optimize the staining, optimize the washing, optimize the various parameters. So it's not as straightforward yet as it may appear, but there is a strong effort on pushing it more strongly in that direction. Another effort that is being done is, of course, the labeling systems themselves. I think it's clear that having 2 of these, maybe 3 is limiting, so we will need more of these systems, and also we will need to develop better understandings of how they work and how that relates to their properties. A very big push that I see also is fluorophore multiplexing, and this is a push that has been going on for a while now. It's not that this just started, but I do see this appearing a lot. And multiplexing basically means here measuring many or more fluorophores at once. And the reasoning is obvious. In fluorescence microscopy, the label is in principle the only thing that you can see. So you can only see as many things as you have labels. And this can be limiting. And this is an example of organismal development, where they look at cell differentiation based on by marking marker genes that allow you to know what stage the cell is in. And what you see here is that actually just marking these stages, you're pretty much consuming much of the visual spectrum. So if you want to add more information there, that is non-trivial. So a lot of the push has been on trying to have more multiplexing. One way is, for instance, to try to keep the halo system, but to add in an additional discriminatory factor, such as lifetime imaging. Also within chemogenetic systems, we see this popping up, this desire to go easily tunable, more broad. Fluorescent proteins, we talked about near infrared, also towards other sides of the spectrum, there are efforts. In fluorogenic system, there are also plans or now investigations into having their distinct orthogonal colors. And at the same time, we see multiplexing going on with these smart dyes to unmix even more fluorophores. This is one strategy. We also had something, a related strategy. And in green to red conversion, similarly, we see strategies to go for multiplexing by taking advantage of the dye properties. Also within super resolution imaging, there often you are spectrally a bit limited. A lifetime can open a way out, or easy different labeling strategies targeted to enable fast multiplexing. So this is something that is very active. Also in terms of biosensors, we see development of single emission color biosensors based on anisotropy, which I feel is making a comeback in some ways. I feel a lot of anisotropy is being picked up again. It's never gone away, but I do feel that there is again a stronger interest driven also by this multiplexing. Other ways making sensors that do quantitative imaging within a single emission band, converting FRET sensors into single FP sensors, extending the spectral range, shifted indicators, say to the near infrared, different combinations that really allow you to have bigger multiplexing. Another something, and this is maybe a bit less generally applicable, is the use of multiplexing of sensors based on spatially segregating the cells. This is also something that has seen a lot of attention, or is seeing a lot of attention. Another field that I think is pretty exciting are chemogenetic biosensors. So these are sensors based on the scheme of bimolecular fluorescence complementation to some degree, which I think many of you will know of. And for instance, this is an example study. There are other studies, but where they took such a fluorogen binding protein and they split it into 2 halves. And then when these 2 halves meet up, they can bind to fluorogen and become fluorescent. So you have an interaction sensor. Now, why do I like this as much? So you can see when they associate and then see when they dissociate. The reason that I like this so much is because this makes use of the fact that this interaction is reversible. This binding is reversible, and this allows you to really have reversible BIF sensors in a way that was not possible with fluorescent proteins. And this, I think, is something very cool that's going to go really make an impact within the life sciences also. But in a way, this has been done before with systems based on luminescence, which, by the way, also are fascinating molecules. But it's something that I think is very relevant now, and where multiple efforts and systems are being applied. Also other schemes, chemigenetic systems, to combine the optical properties of such dyes with sensing applications. This is another example that I actually liked a lot because they use their chemigenetic system to quickly evaluate a big range, well, a comparatively large range of dyes to find the ones that work. And this is really a strength of such systems. If you had FPs, you would have to start cloning and making cells all over again. But with chemigenetic systems, you can make your system once and then add in a bunch of dyes and see which ones work well. Also, combinations with bread, I think, is being picked up a lot again, also in line with imaging in more complex multicellular samples. Then something, and I think this is the last part, and then my talk will wrap up, but something that I like a lot, because it is, of course, one of my own interests.
Peter, it's already 11:20, so...
I should wrap up now.
So how many slides are you still having?
3.
Oh, then it's good. Continue.
4. Yeah. So these are sensors that combine light-induced and chemical behavior, multiple sensing behaviors, multiple smart behaviors. So a couple of those things here, systems that allow you to see active cells based on a combined affinity for light and calcium, super resolution imaging, similar of biosensor activities via a number of different ways. I'll go quickly here. Readout of multiple thread pairs thanks to photochromism, reversible marking, again, by having 2 dependencies. This is a paper where we did quantitative imaging by combining photochromism and sensing. And this is an example where, again, we have 2 smart behaviors being combined that are both environmentally sensitive. So, voila, wrapping up. I hope that I've given you an appreciation of the beauty of chemistry in labeling and imaging. There are many studies that I couldn't highlight here, but I'd like to thank everyone for the beautiful science that they have made. It's really a pleasure to talk about this. And maybe I should also acknowledge funding agencies, voila, who support us. And other than that, my story ends here. And I'd like to thank you for your attention, and especially for a nice discussion, which I hope can still continue for a bit. Thanks.
Peter, thank you very much for your talk, and I think it has been given because you started really from the beginning to the end, and it's a really good overview. I also know how confusing this field is because we have a lot of possibilities, and always try to say to people. Sometimes you get people that say, "What is the menu?" I always say. So if you go to a restaurant, you ask for a menu. And there's 2 possibilities, or you get what is the menu of the day, where you have no way to choose, and some people always ask for that. But I guess that in this particular restaurant, there is only a menu a la carte, and we have to choose the type of labeling, that we need for our particular microscopy technique. And I think that you gave a beautiful and absolutely full overview of what's possible, and I'm quite sure that this will help also the attendees. And I see many thank yous from all the people coming over to you, so please read them afterwards.
Okay, thanks a lot. I hope it was useful indeed.
There's questions in the end, but I think I can really say I think your lecture was really appreciated, so enjoy the thank yous that are now coming. Then there is a here, thank you, really beautiful presentation, exciting, so please, Peter, read them. And because
all right, thank you everyone.
We cannot give you an applause, but the applause is there. And there is one thing, people really liked it, and there's also was the question of Sergi, will slides of your tutorial become available, or if any other form of handouts be available? The sessions are all recorded. They will become available afterwards, so you can listen to the talk of Peter again in your own time and in your own pace. However, I'm not aware when this will be, but from the former online form, this was normally around Eastern. So I think then you can still listen again to Peter's talk, and then see in a little bit of, let's say, when things went too fast, you can stop and look at the slides.
Yeah, and of course you can always get in touch with me directly.
Yeah.
I'm not sure how often I will be online at the FOM. because I actually have an overlapping meeting as well where I'm traveling physically. But of course, contact me in any way, send me emails or whatever. I'm happy always to discuss.
I think, Peter, I will do the same. We keep on checking our chats in the meantime. Maybe we cannot respond immediately, but we'll do a bit later. But we keep an eye on our chats. And I thank you all for your contribution. I really enjoyed the lively discussions that we had. Peter said in the beginning that he would only have a talk for 1 minute, for 1 hour, and we really made a full program out of it. It's also due to your contribution. And I thank you all very much. And I'm looking forward to the FOM meeting now, and we are going over in 10 minutes over to the official opening, and I like to see you there.
All right. Thank you indeed.